In 1952, two scientists sealed a mixture of chemicals inside a glass apparatus and fired electric sparks through it. The goal was audacious: recreate the conditions of early Earth and see whether the ingredients of life could emerge on their own. A few days later, something extraordinary happened. The experiment produced amino acids, molecular building blocks used by every living organism on Earth. For the first time, scientists had shown that chemistry could spontaneously create some of life's essential ingredients. But there was a problem. They had not created life, not even close. For more than 70 years, researchers have been trying to bridge that gap between chemistry and biology. And now, a tiny lab creation called as SpudCell may represent the closest humanity has ever come. Because unlike the famous Miller-Urey experiment we spoke about, this new synthetic cell doesn't just contain the ingredients of life. It can feed, grow, copy its DNA and reproduce. Which raises fascinating question. Are we finally learning how to build life from scratch? Imagine walking into a laboratory and watching something that was never alive begin to behave like life. It eats, it grows, it copies its genetic material, and then it divides, creating next generation. Now imagine this thing wasn't modified from an existing bacterium or any living organism for that matter. Scientists build it from non-living chemicals. That is exactly why a tiny laboratory creation called SpudCell is generating so much excitement across the scientific world. Some headlines are calling it artificial life. Others say scientists have created a living cell from scratch. But is that really true? Let's find out. For years, researchers managed to create individual parts of life. Some synthetic systems could replicate DNA, others could grow, others could divide, but nobody had managed to combine all these processes into a single synthetic system that could complete an entire cellular life cycle until now. Researchers at University of Minnesota, led by Kate Adamala and Aaron Engelhardt, created what they call SpudCell. SpudCell can perform nearly all the steps associated with a cellular life cycle. Previous synthetic systems could reproduce individual parts of this process, but SpudCell brings feeding, growth, DNA replication, and division together in one system. It absorbs resources from its environment, grows, copies its DNA, and divides into daughter cells that inherit genetic information. Cell division has been one of the biggest challenges in synthetic biology. Natural cells use a protein scaffold called as cytoskeleton to organize themselves and separate into two cells. Recreating that machinery requires many proteins working together. SpudCell avoids this complexity. Instead of copying nature's complicated machinery, the researchers found a simpler engineering solution. Proteins accumulate on its membrane, creating mechanical stress, and as the pressure increases, the membrane eventually pinches and splits. SpudCell behaves like life in several important ways, but researchers remain cautious. It depends on a carefully controlled laboratory environment, requires external nutrients, and it is far simpler than even the most basic natural organism. It is like a self-driving car that performs well on test track but cannot yet handle every road. The implications extend far beyond a tiny cell dividing under microscope. The future synthetic cells might manufacture medicines, produce useful chemicals, or deliver treatments directly to diseased tissues. This is a big deal because these cells are assembled from defined components. Scientists may eventually be able to control them more precisely than naturally evolved cells. Some compare this stage to the early days of computing. The first computers were limited, but they opened the door to an information revolution. Synthetic cells could do something similar for our biology, allowing us to not only study life, but also to engineer it. Spud cells could also help scientists explore how life began. The earliest cells were probably much simpler than modern organisms. By constructing minimal cell-like systems, researchers can test how non-living chemistry might have organized enough to grow, replicate, and evolve. Perhaps life does not appear all at once. Perhaps it emerges gradually when chemistry becomes sufficiently complex and organized. SpudCell offers a new way to investigate that possibility. So let's talk about implications. What does this all mean? SpudCell challenges the boundary between living and non living matter. It is not fully alive, but it is more than ordinary chemistry. This suggests that life may not be a simple switch. It may exist along a spectrum. And thus, it leaves us with a scientific, philosophical, and ethical question. If humans create something that feeds, grows, reproduces, and evolves, at what point do we stop calling it a machine and start calling a life?